All right, now we're going to look at the endogenous antigen process, right? So where's that process coming from? So this is where the antigen is created internally within the cell, broken down, processed, and now displaced on one of the MHC class receptors, right? So if I were to draw a cell here, you have a cell here, right? Inside the cell, you have this little peptide, antigen, whatever you want to call it. So this is an antigen that is created. So you remember what antigen was. So antigen is some kind of foreign protein that is recognized by the immune system. The antigen breaks down into smaller fragments, right? And this leads, because of this process, this actually leads to the process of the creation of these MHC complexes. So if you remember, endogenous is involving the MHC class 1. So MHC class 1 involving, so this is all of the nucleated cells, and the specific type of immune cell that comes and binds to this MHC1 complex are the cytotoxic T cells. So let's write that. The cytotoxic T cells. So these are the soldier cells, basically coming in and fighting. All right, so they come in and bind. All right, breaks down, and then here in the actual cell, you have this little structure here. This is called the ER. If you remember, what does the ER stand for? Endoplasmic reticulum. Specifically, the rough ER helps for the generation of protein, and the specific protein of choice that I'm looking for now is these MHC class 1 receptors, right? And so what happens here now, this antigen permeates through this ER. So you have this antigen coming into this ER here. Now you have this little antigen, and it binds to these MHC receptors, class receptors here. So these MHC class receptors now have this antigens bind, and the whole purpose of this in the end is to allow for this to be processed by being displayed on the extracellular surface, right? And so after the further processing, the ER transfers all the information, all these proteins to the Golgi, and then the Golgi breaks off and makes these little microvesicles now, right? So you have these little microvesicles coming in that have this MHC receptor, but also displaying the antigen that is present for identification, right? And then finally, you have the final process of exocytosis taking place here. So the vesicle blends in with the cell membrane, and now you have this MHC complex, specifically MHC1, that's displaying this antigen, right? So remember, this is MHC1 receptor. So all of these is the MHC1 receptor. And now it's waiting specifically for the cytotoxic T cells to come in and bind and recognize to see whether to destroy this cell, whether it's some kind of tumor cell, or to find that this is completely harmless and they can leave it alone. All right?